and welcome to the Amplifying Scientific Innovation podcast. I'm your host, Dr. Sophia Ononye-Onyia, founder and CEO of the Sophia consulting firm, a life science marketing and communications consultancy that was established in New York City with the goal of amplifying scientific innovation. The goal of this podcast really is to showcase scientific innovation stemming from global life science companies prior to and following COVID-19 from the perspectives of founders, CEOs, and other senior executives who are working on the development of transformative, life-saving solutions for patients. My guest today is Mr. Ross Youngs, CEO and founder of Biosordia Pharmaceuticals, a biotech company that is exploring the therapeutic discovery and advancement of hidden microbiome chemistry with its breakthrough technology that can be harnessed for immuno-oncology and immunological discoveries, inclusive of anti-infectives for COVID-19 and other viral diseases. Ross is a serial inventor and entrepreneur who holds more than 75 international patents. His list of awards include a Small Business Administration National Business Person of the Year Award. In addition, he's a 5-time Inc. 500 CEO and Ernst & Young Entrepreneur of the Year, Ohio. On a personal note, Ross and I met in person at the 2020 JP Morgan Healthcare Conference following an introduction from Dr. Daryl Porter, a Biosordia board member. In addition, we share common interest in the state of Ohio, medicinal chemistry, and I serve as a key advisor to Biosordia Pharmaceuticals. Welcome to the show, Ross.
Thank you so much, Sophia. It's such a joy to be on here, and I love your energy, and I'm really excited to interacting with you.
Oh, thank you. It's my absolute pleasure. So I'm going to start with my very favorite question. What is your definition of scientific innovation?
That's a good question because I think it can actually be a really broad definition. Mm-hmm. I like to go back to, let's say, how Edison solved problems. Mm-hmm. Edison was an incredible innovator, but there was always a starting point for what he did. Mm-hmm. We think of him as a great inventor, and he certainly was a great innovator, but there was everything he did had a working model.
Right.
So when you think about that kind of innovation, that's an improvement that can be major. And then when you look at, let's say, straight out of the blue innovation or invention that is breakthrough, that changes the curve in a way. That innovation, that type of innovation is so rare. So when you think about scientific innovation, I think we have to think of continual improvement in methods, processes, procedures, and then the occasional breakthrough. So I love the idea of breaking through a barrier that changes a dynamic and lifts the efficiency or the performance of something dramatically.
Wow.
And I think there's some really good examples in history of how there has been scientific innovation that has done just that. And those are the ones to truly be celebrated, but we can't ignore the incremental.
Right. Right. I think that that's brilliant what you said. I think when people think about innovation, they think it's going to be this drastic, as you said, breakthrough. But I think that it's a stepwise process, and it's about taking the little steps to make things better until we get to that sort of big step. So thank you for defining that. I love that very much so. I might just steal that for myself, actually. So just keep that in mind. And then now, out of curiosity, you've done so much, Ross. I mean, it's really incredible just observing your career in the past few months or so, but I would love to know, what is your most notable accomplishment pre-CEO? It could be personal, it could be professional. I just want to know what it is that you're sort of the proudest about.
You know, that is a good question because I sit back and I think about the various things I've accomplished and done, and I feel pretty good about some of them.
Yeah.
And when you go pre-CEO, that kind of puts me in the definition of when I was working for others and before I started to work for myself.
Right.
So one of the things I was involved in was the optical and video disc business. And it was an early business where people were trying to make compact disc, video disc, CD-ROMs.
Mm-hmm.
There was an interesting challenge in that industry in that a lot of people planned on this kind of price per disc when they got into business.
Right.
But because it was a new business and it accelerated rapidly and there were many players in it, the price everyone planned for products shrank to a third within a year. Now imagine trying to do a budget on something that you had at 300% of what you really have today. That was a challenge. So one of the things, I was part of a team, I was head of engineering, and we put together a plan to increase the value of our product again.
Right.
And the way we did that at the time, I don't know if you remember early compact discs, especially for the music industry.
Yes.
They had black printing on them. And it gave you the song title right on the disc.
Yeah.
We asked the question, what could make that disc so much more exciting, so much more collectible, and so much more revered by a user? So we came up with the first technology to put high quality printed images, full 4-color process imagery on compact disc. What happened, and we didn't know this at the time, we knew it was an enhancement, but we ended up getting 3 times the price for a disc because we were the only ones that could do that.
Wow.
And where there was value in that was in the promotional products or when people were trying to get, let's say, their product recognized as a differentiator. Right. So we ended up creating, implementing, building all the equipment, improving all the processes for doing this. And then we went on to win 17 international awards for the images we created. So it was something I did while working for somebody else. But we had to break through some very big innovation barriers, and it did get recognized by the industry as a excellent execution of a technology that we came up with.
That's wonderful. So you've been thinking outside the box for decades and decades and across multiple industries. That's really good stuff. You should be very proud of yourself, Russ, honestly.
And I enjoy that. I love a challenge, or I love a barrier. In my own mantra of what I want to do to succeed, I have really 4 things. It's better, faster, cheaper, or differentiated. And if it can fit at least one of those, then I want to be involved in it. If it fits multiple ones of those, then it gets really exciting. And especially if it's a breakthrough, a large leap in capability. I love that.
Right. And that, to me, that microbiome mining probably falls into all the categories: better, faster, cheaper, differentiated. So what I would love to do now is if you could provide me with a top line overview of potential or ongoing work that may be related to immunotherapy and or anti-infectives within Biosordia.
Terrific. Well, I think there's a couple things to talk about to get to understanding why we're going to have an impact on immunomodulation, or why we're going to have an impact in infectious disease, specifically antiviral. One of the things we know as researchers is the natural world has been prolific at providing starting points for drugs. But our ability to access the natural world has always been inhibited. The best source is microbial. Microbes themselves have led to more than 50% directly, indirectly, or inspirationally drugs that are on the shelf today.
Right.
So microbes have been prolific. The interesting aspect is science has only had access to less than 1% of the microbes due to the inability to culture those microbes. If you can't grow the microbes at scale, you can't understand the kind of cell-to-cell signaling chemistry they create.
Right,
The
right.
ones they could grow,
Mm-hmm.
they ended up getting therapeutics and other products from.
Mm-hmm.
Our whole approach
Mm-hmm.
was, what if you don't have to grow them? So
Right.
thinking differently.
Mm-hmm.
And the challenges we ran into immediately is, how in the world can you collect a bunch of microbes?
Right, right.
And even in the aquatic environment, which is where we play the most,
Mm-hmm.
you can find them in the aquatic environment, but they're different sizes, different shapes, different densities.
Mm-hmm.
So a community of microbes or a microbiome in the aquatic environment
Mm-hmm.
would be a great source if you could collect enough of it at a quality.
Mm-hmm.
So enough means a volume, dry weight volume of this biomass, this microbiome, this collection of organisms, so that you could actually study it in a lab and see all the chemistry creating, and then test this chemistry against targets, whether it's immunotargets or whether it's infectious disease or neuro-CNS.
Mm-hmm.
So what we ended up doing is we started exploring that space
Mm-hmm.
and why people had not achieved this.
Right.
And really, culturing was the number one approach.
Mm-hmm.
And it was always a xenic culture. So you're taking an organism, isolating it, and growing it to scale. Now, there's advantages to that, in that it gives you a pure culture that provides repeatability for GMP, or good manufacturing practices, which is important. In that situation, that kind of discovery works brilliantly. But when you talk about going to an aquatic environment, and I'm going to sidestep a second because I love the aquatic environment because of its diversity.
Yes.
It is so powerfully diverse. You can have high salt, low salt, high temperature, low temperature, high nutrient, low nutrient, different nutrients, high oxygen, low oxygen, high pressure, low pressure, high temperature, low temperature. You're talking about such an amazing natural ability to create incredible genomic diversity in culture. And it's stable to a certain degree, to the degree needed to explore. And it's not been explored to the extent we now can explore it. So it's so exciting to me. But what we do is we go to those environments, which typically represent 500 to 1,000 species. So only so many niches of a given habitat can be filled. Many researchers that have gone to the aquatic environment can find tens of thousands of species, but in one habitat, not all tens of thousands are active. Only a few hundred are active. So you get a real snapshot of a community of a microbiome. The other really powerful thing is all microbiomes are interconnected and related through evolutionary pressures, and also the species themselves. So an example would be, in our body, we certainly have E. coli. In the aquatic environment, you can find E. coli. The interesting aspect is when you look at the overlap of genes and metabolic pathways to our own gut and the aquatic environment, there's a minimum of 73% overlap of genes and metabolic pathways. So it's highly relevant. So our whole technology, which we've taken years to advance, and advance from generation 1 to now we're at generation 4, and full-scale capability to go acquire these microbiome. But what we do is we choose one to go get, and there's a process to that. Once we go to get it, we take our large harvesting equipment and we process millions of liters of water. Now that sounds like a lot, but if you looked at, let's say, a bay or a lake, you're talking about such a small fraction sample, but it delivers us 1 kilogram or 1,000,000 gram dry weight of material. And we pull it out of the water so gingerly that we don't tear up the organisms or lyse the cells. And then it's an average of 2 minutes and 14 seconds we start preservation. One of the things that's very true about microbes is within 15 minutes, they can start to change their metabolic pathways, what they produce and why. They can shut off processes and turn it on. So that's why certain places to do harvesting of microbes does not yield good results, because essentially you might be collecting sludge. And that's the compositions of degradation. We're actually taking snapshots of the active living chemical signaling as well as the genomics of a microbiome. And that's what's so exciting to us, because from that, we should be able to uncover some huge secrets in cell-to-cell communication.
Wow. I think I don't know what better word to use, but wow. Because Ross, what you showcase is that the brilliance of your background is how diverse it is and how you take that forward thinking. As you said, as long as the technology is able to provide a new avenue, whether it's cheaper, whether it's differentiated, whatever the case might be. What I love that you've showcased, and I'm sure that our audience will appreciate, is just how complex it is to do what you do, but also the endless possibilities that microbiome mining might afford us as a new way to solve some of the problems that we've been trying to solve in the healthcare industry for decades and for years. So thank you for that. Bravo.
Well, I appreciate that. I'd like to add one thing for your audience to think about. And that is direct approaches or indirect approaches. So when we were doing culturing, it was a direct approach. We weren't able to look at all the metabolic pathways. We were able to sample the metabolic pathways, but they're direct to the chemistry as well as having the genomics. So you start to fill in this information void. In our particular case, we're now direct. We've expanded not only the chemical information, but can marry it to the genomic information. When we look at some of the processes that were championed over the last 40 years, combinatorial chemistry was completely indirect. It didn't work out too well for bringing drugs onto the shelf. The issue there is the opportunity to run into chemistry that's not real was huge. When you look at genomics, we're talking about an infinite set of possibilities, especially when you look at epigenomics and you look at the metabolome of the genome of a microbiome. You're talking about virtually an infinite complex set of interactions going on. What I can point out is bioactive small molecules made by nature is actually a finite set. It's not an infinite set. And it's such a fraction of the set of possibilities or the set of what genomics could potentially lead you to believe. That is where playing in the future will be critical. Going for what is truly obtainable and real.
No, I completely agree with you. I don't think you could have said that any better. And now that you've given us sort of the context of your innovation, I'm curious to know, have your policies changed? Like, how have you been affected internally by COVID-19?
Well, when we talk about our operation, we certainly did the work from home. We have a lot of people that are working from home. Those that are coming in now as the economy's starting to open back up, we have our current face mask, which is off-the-shelf face mask. We have our hand sanitizers, and we're cleaning the doorknobs and social distancing. So it's funny, I come into the office now, and I'll say hi from 12 feet away, but we'll have a conference call with 3 people in the office, like as if we're at home. So it has impacted us, and it's impacted our customers. But I'm very hopeful that we're learning some valuable lessons
Oh, yeah.
about what we need to do in the future,
Mm-hmm.
because the odds are civilization will face us again.
Right. Multiple waves of the pandemic. I think that we have a lot of scientific data that are pointing to that. And I think the follow-on question to this is like, how have your corporate social responsibility efforts changed as a result of the pandemic?
Well, I think one of the things that we're doing is we've said to ourselves, we certainly have a responsibility to stockholders, customers, and also the people that we work with, our associates.
Mm-hmm.
So we've maintained everyone in employment. So we haven't had to lay off anybody. We hope not to. We've been very accommodating to those that have a higher degree of fear. If they want to work from home and they can, and we'll find ways to help them work from home. So we've really internalized what we can do to keep the company moving forward, the associates all involved and engaged, while we keep our responsibilities to our shareholders, to our investors. We're trying to be responsible all the way around the line. As far as corporate external responsibilities,
right.
We've recognized that there's needs out there. So one of the things that we did do is we started internally in one of our manufacturing companies a mask project.
Wow.
And we put a team together and we said, how can we help society meet the need of a face mask that is, again, better, faster, cheaper, or differentiated? So we've got a program where by the end of this week, we'll be making our first production prototypes of a new type of face mask designed to do some of the things that no face mask currently does. Again, differentiated and better, faster, cheaper. On our current equipment, we can run 50,000 masks per machine per hour. So we're talking about a run rate that is well above anybody's run rate in the world on a given piece of equipment or capital piece of equipment. So we're ultimately looking at having a very fast run rate. The other thing we've done with the materials, and a lot of people don't realize this about the existing mask,
right?
They were made for healthcare situations. Because of that, some of them are appropriate for healthcare situations, and they may be overkill for non-healthcare situations in many ways. They're not necessarily designed for people to wear for 8 hours at a time.
Right.
They're not hygienic. They're not environmentally responsible. In a material like this, you've got 3, 4, maybe 5 different materials. This isn't recyclable. It's not truly reusable. So you run into issues. One of the other issues we've heard is breathability.
Right.
So we have people that are actually having hypoxia.
Yeah.
Because of the inability to move air in and out of their mask. So one of the things we've done is we've gone to a more torturous path of a single material that's basically hypoallergenic, and it is the kind of thing that has been in skin contact for billions of hours. So think about the materials used for diapers, materials used for feminine hygiene products. We're trying to make a hygienic, one-time use mask at a very, very low cost that is recyclable and biodegradable. So we're trying to advance the line of mask products, and that's, in a way, by doing that, certainly we help our own goals and objectives of keeping our employees involved, our investors engaged, but it also helps with society's need of something that's better, faster, cheaper.
Wonderful, and differentiated.
It is differentiated. We really look forward to telling people what this mask looks like. We've got patents being filed this week. It's very differentiated. But I think from our early response internally, I think it'll be appreciated and used by a segment of the population. You
Wonderful.
never know until it happens.
Yeah, and you don't have a prototype yet, correct?
I do, but I can't show it.
No, I get it. I can't wait. There should be a launch party when you do have the prototype ready, because
there should be a launch party, because I think it's going to be fun. And part of our goal and objective is to make sure we find leverage in its production, because we know what the raw material cost and the speed of production is for this. Mm-hmm. Obviously, we need to pay for the development and pay for the marketing and everything initial in order to break even and make a fair, reasonable profit. But ultimately, we can see masks like this being made worldwide very inexpensively.
That is so important. I think that one of the things that we forget when you live in a developed country like the US is how much a dollar costs outside of the US. I was born in Nigeria, and I can honestly tell you, if you're charging, $2 for a mask, for a lot of people that might be living under $2 a day, that is something that we should keep in mind. And I'm so grateful that for someone like you, when you think about innovation, you think about cost effectiveness, not just in the US, but ex-US as well. So that's very much appreciated. So,
and I thank you. One of our other companies previously was involved in the compact disc industry.
Yeah.
And it created packaging for the compact disc world. And it ended up selling billions of units annually. And It was, many of your audience will probably remember AOL discs coming to them in the mail. Some of that was our packaging. When you looked at the kind of product you got with Dell, HP, Microsoft, IBM, we provided everyone with a disc packaging that was sleeve oriented. So it makes sense that we know how to make that kind of product and rapidly.
That's wonderful. So now let's get back to innovation within the industry, more of a philosophical bit here. So when you think broadly, what are some key consideration factors that you believe will be important for sustaining innovation in the life science industry?
That I'm glad you asked. And here's why. I am really big in team and multidimensional teams. Mm-hmm. And I think there has been a very big push by NIH and other organizations that they recognize how valued this diversity of understanding, bringing together to solve problems, right, how valuable it is. We see them doing it in universities with centers now. Mm-hmm. And these centers bring together all these different fields. And I think that will go a long way, mm-hmm, in helping solve some big, big problems. Because we train scientists, right, to know more and more about less and less, so that they are really within this silo. Yeah. And I find it interesting because I've run into people that have been trained in that silo, right, and I talk to them about being a generalist. And nobody wants to be labeled, but they'll go, well, yeah, I'm a manager of a multi-dimensional department, or I'm head of all of research of major pharma. Right. Of course I'm a generalist. No, right, you're not. Right. And the only way you can become a generalist is listen to people outside of your field of expertise when it comes to the bigger problems. Mm-hmm. And I think that all of us are trained with a set of knowledge. Mm-hmm. And it's really difficult to be able to touch so many areas. There are some people that can do that. Mm-hmm. But I think that there was a Harvard study, and it can be found in the Harvard Business Review, and I can provide that link to you. Okay. But it talked about where innovation comes from. Mm. And innovation, 50% of it comes from specialists. Mm-hmm. 50% of it comes from generalists. Mm-hmm. I'm not saying there's not validity in having specialization and understanding a subject highly. I think that's critical. Mm-hmm. But understanding how to use specialists so that you can turn a team into generalists, mm-hmm, is huge. And we always have the dynamic of team dynamics. And there's always the ego involved. And group dynamics within innovation is critical. And it's not used to the extent it should be used. And that is, everyone should come into that group fearless, completely fearless. And everyone should come into a group that's trying to innovate for success with respect for all of the opinions and avoiding leadership outside of organization. So organizational leadership is fine. Thought domination is wrong. When you're really trying to solve the big problems, you have got to have open minds. You have got to play with the whiteboard as it really is a whiteboard. And I think that's when and how these bigger issues get solved. It's not one mind. We see often in science, one mind is brilliant.
Right.
But I'm here to say that that one mind, calibrated and used properly, could be beyond brilliant.
Yes. Once again, I completely agree with you. I think that diversity of thought and being able to recognize and appreciate an interdisciplinary approach to drug discovery is one that we should be very appreciative of. And I think if there's any positive start, like when you think about COVID-19, one of the positive implications is this idea of collaboration as we all continue to come together to think outside the box. So that's really great insight that you just provided. Now, just out of sheer curiosity, because I'm a curious person, is there any technology or company that you're currently excited about? It could also include yours. I just want to know what gets you excited.
Well, you know how excited I am about Biosortia. There is just no doubt about that. I've thought long and hard about the companies that excite me these days, what kind of innovation, what kind of opportunities they have. And I am an investor, so I get to see some different companies. And it may not be fair for me to label one as too exciting. Historically, we've seen some really good successes come from technologies. And I think we've seen some really good successes come from life sciences. I'm disappointed in life sciences' lack of, in general, risk-taking. Many industries have been taken over by the financial people. They lose strategic vision. And I think that that's dangerous because there should always be risk taking. If you're not taking risk, you're winding your company down. And if you do not find opportunities to provide thought leadership of some kind in a field where you can excel, where you can motivate your associates, your employees, your collaborators to have a vision and embrace that vision and understand the strategy, you're already failing. And there's too many companies that are in that mode. And there is so many opportunities to excel. And it's all about the numbers. And it's all about, let's reduce our risk, let's leverage our risk elsewhere. And I think the biggest winners come from internal focused creativity, not from waiting till someone else does it and buy it. Because quite frankly, they're already selling that company probably for too high of a price. And I think that for companies to ignore the opportunity to find targeted opportunities with a strategy and a vision is a huge mistake.
No, I agree with you. I think that the risk taking is probably something that I feel very strongly about as well. I wrote about it in a piece in MIT Technology Review, and I shared that that's probably the reason why we're not as prepared for COVID-19. And so
Exactly. Exactly.
So let's come up with this hypothetical scenario, right? So there's a new CEO, their first day in the office is today, a new biotech CEO. How would your advice be different today for this new CEO versus pre-COVID-19?
Wow. Oh, you are so thought-provoking. You know, things have changed.
Yeah.
And there is that recognition.
Yeah.
I think life science in itself is such a broad category that even for that CEO, they've got to say, do we have an opportunity? And the opportunity should be thought of two different ways, because there is the opportunity to profit and there is the opportunity to do the right thing. I believe that if you're doing the right thing, profits will come.
Absolutely.
So I think they should be thought of together. And I think if you're a new CEO walking into a company and maybe you understand some, I think you have got to talk to your main leadership team in an open forum where you can get comfortable talking about what we should and shouldn't be doing. I think the first thing you do as a new CEO into an environment like that is you eliminate all risk of that executive team. If that executive team has a hierarchy where we don't talk, it's only that person that talks,
right?
That's got to go immediately.
Absolutely.
And I think that the challenge is there's always those egos. There's always that dimensional stability to an existing team.
Yeah.
And you have got to rough that up and create a freedom and a willingness to engage. And that has to occur in open forum where they feel safe. Doesn't mean there isn't side conversations going on. But in the side conversations, you have got to get rid of the pettiness and you've got to create a team work in order to find the strategy, let alone execute the strategy, but to find the strategy and the vision.
That's really great advice. Honestly, I'll take that advice as well, honestly. But I'm going to give you the floor for any final commentary or thoughts. Anything you really want to share that you haven't said already, you have the floor now.
Well, I'm a believer, and again, I think we're spearheading this entire technology revolution.
Yeah.
I am a believer that prospecting or mining the microbiome for its hidden secrets will be something almost every research group 15, 20 years from now is involved in some way.
Wow.
I think it is such a wide open space to connect the actual chemical signaling to the genomics, to the activities, that this is going to really open up opportunities to have next generation therapeutics. But it doesn't stop with therapeutics. It goes into agricultural products. It goes into longevity. It goes into consumer cosmetic products and sustainability. So to me, having these secrets here and accessible is different. They weren't accessible. We only had the indirect methods. We're here to say we can directly interrogate this hidden chemistry. And the fact that we're not, it's malpractice. It's absolute malpractice.
Right. Right. Wow. Okay. Well, Ross, I can't thank you enough for just all the insights that you've provided. I think my biggest takeaway was about how when we're thinking about innovation, it should be better, it should be faster, it should be cheaper, and it should be differentiated. You've taken a very complex subject matter like microbiome mining, and you've done it with a smile on your face. You've basically broken it down for anyone to really understand what potential this technology has. So I appreciate your time, and it's been just amazing speaking with you today.
Such a pleasure. I love your energy, and keep this up. Keep spreading the word, and it's such a pleasure to be involved with you, work with you, and engage you. So thank you.
All right, thank you, Ross. Have a good one now, okay?
Thank you.
Bye.